harmonizing Benchling data with LabKey. So sort of a follow-on to Katie's presentation. Actually, I am going to start with a bit of information about two technologies. Katie has already mentioned one of these technologies, referenced external data sources. So I'm going to just describe that in a little more detail, and also going to describe a facility called ETLs within LabKey. So these are, as I mentioned, we're going to talk a lot about data integration. This is obviously one example of that. So these are two technologies that some might not be super familiar with. They're sort of built-in capabilities of LabKey. And I want to give credit where credit is due. That is one of our developers, Binal Patel, put together sort of a longer version of this slide deck, and I stole all of that material from her, but want to make sure that she gets credit for it. All right, so as you heard, LabKey is able to connect to external databases. So there is a primary LabKey database, kind of the LabKey database, where a lot of the administrative data and assay data, data sets, lists, a lot of the data that goes into LabKey is in this primary relational database. But LabKey can also connect to any number of other relational databases within your institution through this external data source, or sometimes we say external schema mechanism. You can see down below a wide variety of different database servers are supported. You've heard about Postgres, but we also support SAS and MySQL and Oracle, SQL Server, etc. And so what's an example of using an external data source? Well, let's say you collect clinical study data, and that database of record happens to be SQL Server. Maybe you collect demographics data in Oracle, and you collect all of your assay data from instruments and process it and import it into LabKey. An external data source basically allows LabKey to connect all of these databases together. So that looks something like this. LabKey uses a mechanism or an API called JDBC, and a JDBC driver that is specific to each database type, in order to kind of get a generic view of these different databases and to provide access to those who have been granted permission to see that data. So that looks something like this, where an administrator chooses to expose certain schemas, just like Katie showed, within LabKey, within folders within LabKey, and secure those folders. When that's in place, a variety of facilities within LabKey can be used to view and analyze that data, to sort and filter and create statistics, to create charts and visualize that data, to use APIs to create applications, say in JavaScript or Python or Java or some other language, to create custom queries and reports on top of this data. So a lot of the kind of the key features of LabKey can be used on top of each of these external schemas. So I've kind of gone through all of these already. I guess the main thing here is just to show that this can be set up very quickly and easily. Just a small bit of XML goes into your LabKey.xml file so that LabKey knows what URL to connect to, and provides a set of credentials to use when LabKey is connecting to that database. And then it's just a matter of selecting that database and choosing the schemas and tables to expose within LabKey and in what folders they should be exposed. So very quick way to connect LabKey to any database within the institution. Now, there are some limitations. First of all, those different databases, the LabKey database and these external databases, they're all fairly independent of each other. You cannot write a query that executes a SQL join across those different databases. They are kind of separate from the standpoint of SQL queries. Now you could write some JavaScript or code in another language to combine data in different ways to sort of do your own joins within code. But in terms of a SQL join, that's not a possibility. Also, it's worth mentioning that these schemas, they're not true LabKey data structures in the sense that it's not a data set or a list, doesn't have those sort of special study or built-in LabKey features when you use an external schema. So to look at this visually, those X's are kind of showing cannot cross these lines, can't join between these three different databases that are kind of shown here. So there is another facility that can be used to kind of get rid of those X's. This is our ETL, or extract, transfer, load facility, that essentially in this scenario is just being used to copy that data from an external schema into a data set within LabKey. So in this case, we've used an ETL to copy this clinical study data and the demographics data into corresponding data sets within a LabKey study. And with that in place, I can access that data in the data set structure. So the ETL built into LabKey is very feature rich, lots of different capabilities. This is really just at the very, very high level. You can run an ETL manually. You can run it on a schedule if you want. You can customize the ETL to append or merge or truncate or re-import. It provides for incremental updates if you want. And you have a number of different ways to provide this configuration. You can define the configuration of each ETL within a module, which can be helpful if you want consistency across different servers, say a test staging and production server, or you have multiple developers working on ETLs. That's a very handy mechanism. Or you can provide them and edit them within the LabKey user interface and just store those configurations in the LabKey database. So with that in place, with an ETL in place, you can kind of see here, all that data is now stored in the LabKey database. It can be joined across those different data sets. The apps will have a simpler time of joining that data. You can write queries, reports, all across the combined information, and you can take advantage of all the study facilities that LabKey provides. Okay, so with that background, with those technologies kind of described, I'd like to introduce the panelists for harmonizing benchling data with LabKey. We have three folks joining us. Jacob Jaffe, head of data science, also at Inzen, which is where Katie's from. Huang, scientist at Regeneron Pharmaceuticals, and Mathew Hoops, scientist at Adicet Bio. So if you folks would turn on your videos and unmute, we'll get started with some questions. All right, so I'm just going to kind of go through a few questions here that I have. Actually, you want to just test out your microphones, make sure we can hear you.
Check, check.
Check, check.
Yep, sounds good.
Hello.
Excellent. All right, well, why don't we start with you?
Mm-hmm.
So can you just kind of briefly describe your general research focus and the challenges that brought you to LabKey, just in general?
Sure. So actually, I'm from analytical chemistry lab. So it's actually a development lab. So we actually characterize different of our product, mostly antibody. It's not like proteomics. We characterize it, using mainly, primarily mass spectrometry. So most of our data are mass spec data. So, and then, from like, I think it's 2017, we got a chance to see the Panorama partnership. So, we also use Skyline to process a lot of mass spec data, mainly targeted mass spec data. And then, we feel that, okay, so the Panorama and also the LabKey server can help us to host the data to better organize our assays and, to host all, and also, we're also thinking of, putting our historical data inside. So, we definitely need a data management system, and, it have to, accommodate a lot of mass spec data. And at that time, and we actually searched around, and, the Panorama and LabKey is the only solution that, we can host the mass spec data, in terms of extracting ion chromatogram and the libraries. And also, you can see the spectra, you can see the the tandem spectrum match. So at that time, this is the only solution, and we also use a lot of we actually all use Skyline to process our data to do the quantitative analysis in terms of the attribute analysis and also the whole cell protein quantitation, or even some of the absolute quantitation. We use Skyline. So, Skyline is best integrated with Panorama. So that's why, we actually use mostly use Panorama here. Use the Panorama module. And so the reason that we are integrating Benchling is that, our company store a lot of like molecular entity information into the Benchling, and also the sample request information, sample metadata, and also the task distribution information are all in the Benchling. So, and we because the sample we're running on mass spec, they are from the sample information from this source. So we're thinking, we can connect this information, so we we can better serve the downstream analysis. So we actually couple the LabKey with the Panorama. The spot fire. So the factor, if we are getting metadata, including some of the experiment condition factors, so it better help us to visualize the data, to do the further analysis. So that's why we are we are integrating the the sample metadata information from Benchling, and also the some of the molecular identity or molecular calculation into the sample. It's a sample relay, sample information in relay to the Panorama server, so that we can have the data, and then the data can be further relayed to Spotfire for analysis and visualization. So that's how we use this, and how we connect with Benchling.
Okay, great. Let me turn to Mathew and kind of ask the same sorts of questions. So first of all, if you can kind of describe your general research focus and general challenges that brought you to LabKey in the first place.
Sure, absolutely. So Adicet Bio is a CAR T cell therapy company. And so as a cell-based product, one of the key instruments that we use throughout the company is flow cytometry. And LabKey was actually introduced to Adicet first before Benchling, and then throughout the development for the analysis of the product, as well as the research into various aspects of cell biology, flow cytometry is just central to everything we do. So the module in for flow cytometry in LabKey really was a bedrock of where we started. And as a young company, we then later added the ELN Benchling to our laboratories, the various laboratories. I myself am in the protein lab and a member of the molecular engineering group. And so I started out storing data about protein entities first. And I think what we've found in the company so far is that we've really developed a sort of Kimball style data mart approach to our Benchling data. And one of the things that we learned was that as these sort of different data marts were evolving from different groups, that we needed a way to tie them together and also integrate them with our analytical development with flow cytometry in LabKey. And then we began exploring ways to, sort of leverage more of the resources available in LabKey. So that's really the big picture. And really with the product like cell therapy, you've got viral vector, you've got protein reagents, you've got the donors from the cells all going into the final product. And people wanted to ask questions about any one of those, from the sequence in the car all the way through, the vector lot, the donor, various reagents that went into the process development settings, everything going into the final product, and what might be analyzed with respect to that part of the product. So that's where we started developing all of the different entities in Benchling, which are essentially provide the framework, the interlinked framework on which all results can be attached, and then bringing those into LabKey and continuing to do the attachment with the flow information. So that's the framework against which we are doing our development in LabKey. And so that's an ongoing project for sure. We have a good team that helps out from different departments.
Right. So it sounds like entities get registered in Benchling and the flow cytometry data resides in LabKey. Is there other significant data in either place, or does that kind of capture the primary data?
Well, the entities are key, and then the experimental conditions, it's an ongoing process, but theoretically they could be either place. Oftentimes, because we have ELN templates that manage the collection of information for, specifically for process development, it's a slightly different scenario, but parallel for some key research areas. But one of the things we're learning is that, we're leveraging where they're the same and where they're different, and looking at things that are more stable in terms of, historical repetition of use, and pushing those forward. And then as we learn different ways to use the products, then we apply them to, edge case scenarios, for instance, in research that might be able to use them as well. No, but to answer your question, primarily it's entity and some results in Benchling, and then aggregation and some additional results in LabKey, and then visualization in LabKey is where we're looking at right now. And so that's where the process. So, the diagrams you have shown about JDBC connections to external sources has really been something that we've been thinking about a lot in recent times. Yeah.
And so just touching a little more on the reporting or visualization, either that you've done or that you expect to do in the near future. Mm-hmm. Just sort of describe what you're looking at in terms of data analysis on these combined data sets.
So in the past, we had someone who was an expert in our reports, and so we've done that a lot. As we continue the migration, we're looking at other possibilities, and that may be a key part of what we do as well in the future. One of my contributions is really just to make sure there's no unbroken links between entities, mm-hmm. such that whenever you visualize something, you can always drill back down to say, hey, where did this data come from? Obviously, that's key. Mm-hmm. So, in the way I think of all of the SQL and ETLs that get data from external sources into LabKey is that it's traceable, and you have that ability to go back to the source, but then you really have people who want to ask questions of the data sort of in natural language style, and having the people that are helping out with bioinformatics and data sources be able to translate that consistently into those kinds of analyses, I think, is what's key.
Right. Yeah, it sounds like that provenance or chain of custody piece is very similar to what Katie was showing.
Exactly. Yeah, for sure.
So speaking of Inzen, Jake, same kind of questions to you. I know Katie kind of covered a lot of this already in her talk, but we'll let you say it in your own words. So again,
yeah, I'm not
research focus, challenges, and what data resides where in your case.
Yeah, so I'm not going to rehash the story of Inzen and our focus on cytokine biology, but I will say I came into LabKey through an unusual back door. Before I was at Inzen, I had been at the Broad Institute working in the proteomics platform for many years, and I was really familiar with all the things that LabKey could do for proteomics. I was a PI of a large grant there. You're going to hear from my collaborator on that grant, Mike McCoss, tomorrow. And we really relied on the proteomics aspects of LabKey to be able to share our data there. So fast forward, and I show up at Inzen as head of data science, and it's just me and one other person in the group. And I'm thinking, gee, I don't really want to have to stand up relational database and manage all that kind of stuff, and started looking into different vendors of kind of solution providers. Then a sort of light bulb went on in the back of my head, and I was like, oh, that LabKey thing that I used to use in proteomics, it can do a whole lot more, can't it? And so then started talking with Bernie and folks at LabKey, and pretty quickly came to the conclusion that LabKey was the right solution for a small data science shop to be able to hit the ground running. The thing that really kind of sold it for me was the ability to get native computable objects through APIs for several different languages that were available without having to go through hoops and coercion and all kinds of weird stuff. So and it was important to us that it could connect to external data sources like Benchling. You've heard how our goal is really to meet the scientists where they are. I love Katie's phrasing of that. Benchling was a well-established thing here at Inzen, but before I came, and so we wanted to meet them where they were. And in terms of the data that we chose to keep in Benchling versus things that we put in LabKey, that was actually a really important discussion with the folks in the lab. We said, hey, what can you do in your day-to-day in Benchling that's going to make your life easier versus asking them to kind of do stuff in LabKey. So that's how we kind of got there. And I'll say we kind of use LabKey in all those ways, both as a sort of aggregating single source of truth between experimental metadata and the actual data that comes out of it. We run ETL jobs even on our own thing to create summary queries for fast display of information and progress. And then we do use it as a backend beyond the built-in visualizations that we can build in LabKey. We use it as a backend to stand up Shiny applications and stuff like that. So it's really like the central tenet of the data science group here at Inzen. Yeah, I think that pretty much sums it up.
Nice. So I know Katie showed examples of using R against the combined data sources. You mentioned there might be more than one language that you're using. I'm just curious what other languages and tools, like you've obviously mentioned Shiny already, and so obviously R, but
right.
what other languages were important?
Yeah, we had a sort of summer intern developer use the JavaScript API to stand up some functionality within the LabKey server itself. We've done some stuff with Python when that was necessary for joining to other kinds of processes that we have going on here. So the shop itself is heavily R, although is basically language agnostic. We use the right tool for the job.
Great. Yeah, and as I mentioned earlier, we try to do the same thing. LabKey be fairly agnostic and provide APIs and bindings for a variety of popular languages. Before I go back to you for a couple of minutes, I just want to remind participants there have been a couple of questions that have been added to the Q&A chat. Feel free to add more if you've got things come up, if you've got questions for our panelists, please just type them in the chat window. So back to you for a minute. So can you kind of describe technically how your integration was set up? What are the different components or LabKey features that you use to integrate this Benchling data, and how does the data flow from Benchling into LabKey in your case?
Okay, I think so. I think this first start is from communication, because I'm a lab scientist, so I kind of work with the system and the things I need to contact with our lab IT and and also our lab IT, that they know how to manipulate some of the query and also write some queries on the benchling side, because the our benchling is has a lot of information there, and they have very customized query, and so on that side I contact like several internal IT people, and then on the LabKey side I contact with account manager case, and and I think I forgot exactly who, but the things I contact them, and then set up a meeting, because for me it's kind of, I'm a scientist, I mean for me it's like kind of I don't know which to connect, it's probably those codes and those port something, but eventually we are able to connect all the people, and I feel that it's some configuration in the LabKey XML, and you specify the the location of that, the web, I mean the port, the location for that benchling. I forgot, for that benchling we definitely need to create a account, the account to connect the benchling and also the LabKey, and then we use that account to connect this it's from what the the Benchling warehouse. It's a account for Benchling warehouse, and we have that, and the through the configuration, I think the first thing we establish external schema in the LabKey, so we can read all the schema from the Benchling, and then I think because the Benchling information there is a lot, so we only expect some of the information. So our internal IT, they wrote a the query that specifically extract the the sample information or the sample metadata, some information that I need, and then have a customized query. And then I think we have the query, we have an ETL that save the information to the internal list. So we actually pass the information from the benchling and to the list, and then we can join the list. We use some key key identifier for samples to join the samples that we have on the LabKey to the benchling. So I think then I get more feeling about how the things are connected, how things can be like updated, and how all the dots are connected. So yeah, that's my feeling for the the connection.
And how regularly does that ETL run? Is that a nightly?
1 day and every day, 24 hour. Yeah.
Yep. Right. And did you encounter any challenges when you're setting up this integration, or working with others to set up the integration?
Yes, I think no, there's some, like, because we are actually it's not like appending data, it's because we are like taking the the identity data, and then I think the first few days we didn't know it's like I think the default is appending, so there are a lot of repeat of that identity, and then when we are joining the information from the metadata with our data, there are some errors, and we see. So then we change the how the data is acquired, and it's always refreshing new it's kind of refreshing new data, and then we can every time we can use that identity to join the new data set. So that's the thing. Yeah.
Okay.
Yeah. Other thing, I think it's working pretty good. Yeah.
Great.
Mhm.
Yeah. So it sounds similar to what Katie was describing in terms of important to validate the data as it comes.
Yeah.
To make sure it's what you expected, and if not, obviously adjust it. Are you expecting any kind of follow-on work, or is there a vision to take this and further, or are you kind of set with what you got here with this integration?
Yeah, I think there was some problem with like the JSON blob. Yeah, I forgot. I wasn't too much familiar with the technique. So there's a because the benchling support all of the other formats, and when we are importing the data, there are some fields that I think we ignored because something that we cannot handle at that time. So yeah, so definitely I think we're looking to integrate more information from our benchling, because that's our go-to set for the assay, the molecular identity, and also the sample, all the sample information. So, and actually our benchling are integrated with our ERN already. So which means we can extract more information from there, to pass that information to the mass spec data we have. So and I think that some difficulties are we need to regulate, or not regulate, harmonize more, or get some restricted fields, so it can be better structured when we are inputting the data in the benchling side, so it can relay all the structure information to the LabKey. So
Great. So I'm going to take one of the questions from participants at this point. So the question was sort of a general one, and it goes to what you're just talking about. How does one connect an external database with ETLs? And I can, I'll just go ahead and answer that because it sort of fits in with the presentation I did earlier, plus what you was just saying. So basically an external database gets connected, as we've been talking about, as using this external schema mechanism. So that schema is then made available in a particular folder. Again, it's secured using all the usual access control mechanisms. So only those who have read access to the folder can read that schema. And then ETLs are configured. Oftentimes, their job is really just to copy data from one table or query that LabKey can see is exposed to LabKey to another table or query that LabKey can see. So in the case of, in use case, he mentioned setting up an external schema. That schema is now available. There are certain, there's a table or tables within that external schema. You could even write a custom query on top of multiple tables, but that table or query is then accessed by the ETL. So the ETL literally is configured to say, go to, the benchling schema dot, samples table, let's say, and copy the data into list dot samples. So the list called, with the name samples, and configure the ETL with that and set it to run, say, on a nightly basis. And every night it will simply query the data from the external data source, which is that benchling database in this case, and copy that data into the list. And as you was talking about, there are options to do this incrementally, or to truncate and replace, or to append. It just depends on how the data are coming in and how the particular database that you're talking to is configured, or how the query is configured. Because of course, if you have a query on top of this external schema, you can make it filter, out so that only the newest rows are showing up, or whatever you need. So that's the basics of how external databases can interact with ETLs. And the good news is this is all documented on our public website, so you can go to labkey.org and learn much more detail about both external data sources and ETLs. All right, so I'm going to go back to Mathew with a few of these same questions. So can you just kind of briefly describe how you set up your integration and the LabKey features you're using and how the data flows through that process?
Sure. So the once that external schema is set up through a data reader account from Benchling, then when the developer looks at that external schema and wants to select tables to write a query, they essentially have, they can see all of the tables in the warehouse. And so I think one of the things that we found is useful to do is to select the tables for the particular purpose that you have in that particular folder and write a query that combines them, and then ETL that query over into your lists for native lists in LabKey. And then you might do that a few times for whatever particular purpose that folder serves. And then you can finalize any kind of query and visualization scripts from those native LabKey lists. That's essentially the process that we have now. Very similar to what you're describing. But I mean, I think, as development continues, there's always an opportunity to find ways to smooth out how the data gets to its final destination in LabKey. And oftentimes that can be a small tweak in the SOP that a bench scientist is using for how a template is used in Benchling. And we have some very good Benchling administrators that are able to go and add a field to a result table, because that allows that data that's in that result table to become more widely used for multiple purposes because of the link that new field provides. But it is important to think carefully about setting up results tables, because they can be somewhat sticky in terms of once the tables are established. So that's one recommendation. Think carefully about your results tables, but really focus on working on the interlinks between entities, then your results tables. And then when you expose your warehouse through the external schema in LabKey, you really have a great base of information off which to work in what you pull over. And then for the things that you put directly and natively directly into LabKey, and like I said, we have flow data that comes in that way, some Luminex data that comes in that way. And really the question then is, once everything's normalized to the internal LabKey schema, then they will play quite nicely with each other. I think that it's really great to see a table or a plot, and as a developer, where all of that data originated and see it all right in front of you. And I think that's what the user imagines in the end, is that they have data that appears in front of them the way they work and think.
Right. So you hinted at a best practice there of really thinking about columns and that kind of the structure of data, I guess, because it's tricky, because reports and queries and other downstream analysis ends up getting built on top of those
exactly right
elements. Any other best practices or any challenges that you encountered in that integration process?
I mean, I think it's always instructive to stay in close contact with the users and sort of shadow their work on occasion or frequently if necessary. I've learned a little bit about how sample submission occurs in the people that are working in process development and analytical development, and when flow samples are submitted, what's been done with them prior to that, whether they're stained and run immediately or whether they are fixed and held for some period of time. And I'm thinking about what fields need to be available to the LabKey ETL process or somewhere just before or just after that in order to validate that, a hand-entered timestamp versus a database auto-generated timestamp are useful in terms of how you manage the the validation of what's coming across, especially anything that's automated. And then also having some trying to design some processes that have manual fallbacks so that when something's under development, it can be tested manually, or when it's automated, it can be monitored, and if some development is put on pause, and that there's something that the user can keep doing. I mean, that's a kind of a broader view of development, I guess, but it seems to be something that I find useful.
All right, and Jake, can you kind of remind us how Inzen set up this Benchling integration in your case, and the LabKey features you're using and the data flow?
Yeah, so for the Benchling integration to LabKey, we do not use an ETL to move stuff over. We don't relocalize the data from Benchling onto LabKey. And my design philosophy was I didn't want to have the same data in two places. I wanted to kind of leave it in one, but just manifest it in the other. So, I'll say that our LabKey instance is hosted, and so it was really easy to set up this integration. I just spoke with our kind of support folks at LabKey, and some tense moments exchanging the credentials or whatnot, but yeah, that was pretty turnkey from our support standpoint. Other than that, we do make visualizations against it in our LabKey instance itself. And as you saw, we use the LabKey API to reach back over into Benchling in order to do the really important validation work before we let our pipelines run forward. And actually, I remember there was a question during the live session. Does this happen? Does data push or data commit happen in real time? And the answer is it does. It's the final artifact of the processing pipeline is to commit the data to LabKey. So whenever the pipeline runs and generates its kind of final artifacts, they do get committed into our LabKey server. But we know, like others have mentioned before, the data provenance is there. We're going to be able to trace the results back to the entities, back to the notebook pages where the original samples were generated.
So there was actually a question by somebody who knows some of these details. Can you kind of comment about? I think you brought this up as well, the JSON entities and support within LabKey SQL for those, and what you found there, what you used.
Yeah, I mean, as you had alluded, Benchling does some things in their data modeling where they choose to manifest certain types of fields as JSON entities, whether it's like a multi-select box or some such thing where controlled vocabulary is desired, right? The actual field types that get populated in their warehouse are of this kind of JSON or JSON-B type. I realize this is pretty technical, but like at a higher level, it's kind of the relational database's way of cheating and trying to also be a no schema database or a schemaless database, to have fields that are essentially sample records in MongoDB speak as little fields in their relational SQL database, right? So it's actually good. It's really flexible. We use this trick ourselves. We use this trick in LabKey. We manifest JSON type fields in LabKey. They manifest as strings. So we were kind of having this problem with being able to parse things from the Benchling warehouse, plus we wanted to parse our own JSON record-like entities in our own data models. And so I guess what I can say is, talk with your LabKey support folks, because we talked about this again and again and again, and they're like, ultimately it got done, and now LabKey makes some very key JSON operators available as part of their dialect of SQL, which enables us to now natively reach back into the Benchling warehouse and deparse things as we need to. It lets us use our own tricks to deparse our own kind of sample records that we use in a really flexible way. And so, to that extent, it's a useful extension of what LabKey could already do, and plays nicely with some of the more modern evolution of the kind of core SQL backends.
That's great. And I guess in your integration, any particular challenges that came up? I mean, I guess the JSON.
The biggest one that we had was the JSON. The JSON thing. I think there's some other funny things about how Benchling manifests their data model that uses some funky characters and field names that I think has continued to be a challenge, or at least we haven't checked recently.
Well, I think that may have been fixed. At least I hope it has, because I investigated that and hopefully fixed that particular problem. Yeah, that had to do with escaping of certain characters within query names, within like operators. It's a little obscure, but I think we've got that going now. So, and then I guess final question for you, Jake. Any follow-on work that you expect, or is there a ultimate vision or direction you want to take this work going forward?
I think what we're working on now is really expanding the types of data that we ingest directly into LabKey, while maintaining that kind of connectivity back to Benchling. And what we can do with both the Benchling and the LabKey APIs to make that a pretty seamless process, keeping it pretty transparent for our lab users at the end of the day. Great.
I know there was another question from participants about specifically Plotly. I think Plotly and our LabKey kind of combined. I'll just chime in a little bit on that, and then if any of our panelists have experience, we'll move to them. But so we added a few releases ago support for Plotly Dash, which is more a Python-based Plotly facility, so that you could potentially within a LabKey portal page or within a web part put a dynamic Plotly Dash app, or output from a Plotly Dash app, and have two-way communication between data in LabKey and Plotly. I've only experimented with that with Python. I haven't experimented with that with R. We're talking earlier about Shiny, and we've added integration with Shiny just recently. Same kind of mechanism where you can have a Shiny app that then is essentially proxied by LabKey for security purposes, and provides kind of sharing authentication, which is helpful and more seamless, and having that output appear within LabKey pages. So I guess any other comments on that, or experiences that our panelists have had with either Plotly or our LabKey and Plotly Shiny?
Yeah, we've used the Plotly Dash, and that's been very nice to see that dynamic visualization as well. And we hope to leverage that further in the future. We're just getting started with that, so we appreciate that work.
We've just done some basic, standing off of Plotly plots that are not necessarily data connected. They maintain the Plotly general interactivity in terms of zoom and highlight and stuff, but not in a data connected way.
All right, well, I'd like to thank all of our panelists today. In fact, I'd like to thank all of the speakers today, and thank all the participants for joining us. We are a couple minutes over, but I hope this has been a valuable experience for everybody, and we look forward to seeing all of you back again tomorrow.